So we have heard over the past day and a half a variety of presentations looking at models of biology in different contexts, and we've seen a very sort of a number of shared themes emerge repeatedly, and I'm sure we'll be touching on those in this discussion. But if I may, I would like to start with a question that really is kind of keeping me up at night these days. And I'd really love to hear your perspectives on it. And that's, so let me set the scene, as it were. I think we all agree that the human immune system is immensely complex. It's very large scale, trillions of cells distributed all over the body, doing different things in different compartments. A large proportion of the cells are dying and being replaced all the time. We're constantly being challenged environmentally and responding. And we know that the immune response, as we've seen in the presentations, can range from adaptive nonlinear responses that go from seconds to decades. So that's the context, and in that context, we're increasingly collecting more large-scale and more detailed data, right? Everything is single cell. We're looking at individual molecules in the case of Harlan's presentation. The question that I want to pose to you is, what do we do to make sure that we're not, to analyze all this data, we have to use very complex mathematics and statistics. There is no choice. There's very complex transformations that happen to go from raw data to meaningful numbers, and then further mathematical transformations that we impose to make systems-level models. How can we be sure that we are not imposing our own wish fulfillment on the data? We have so many parameters, so many mathematical transformations. How can we be sure that what we're getting at the end is an unbiased representation of the biology? Does anybody want to go first? Martin?
So I think I have a microphone already. So, I think that that disease states, pathogens, or cancer, they are actually already doing part of the job for us because they're probing into what the immune system does. And when we look at those cases where the immune system fails, the molecular players that pop up are those players that are most likely to be important. And so the defects of the immune system, as we can correlate them to defects in, for example, molecular expression, actually show us where to look. So, for example, the TCR variability. So if you look just for a particular TCR somewhere in a population of people, you pick Grégoire and you pick Shai or Catherine, and you say, okay, here's one TCR specificity. Can I find it in another person? Just looking for it, you won't find it. But if you look for a certain pathogen specificity, then and you know that both people have been exposed, then suddenly it pops up because the immune system has been probed by a disease, by a certain condition. And I think, this has been, for very practical reasons, the guiding principle over decades of research. All the important molecules that popped up over and over again became important in publications and research because they became important for certain phenotypes that we observed.
Does this thing work? If I can add to that, I can tell you from the perspective of the TB field, where we're very confused about the immune system, that one idea that we're coming around to is that we can use perturbations and the pathogen itself as a readout of the true outcome, right? So to not be confused by the instances of these different molecular players, but what is the end result? And so one way to do that is to use, in our field, the collaborative cross mouse systems, where you get natural variation, so to move away from the model animal systems, and then to use our advantage using a pathogen that's slow growing, as we can get some that we can introduce reporter strains, and so that we can use that as a sensor, use the bacterium itself as a sensor. You know, we show that we get differences with drug, but they adapted to the immune response. So the idea is that we can look at the bacteria themselves to tell us what stresses they've been put under with these different animal systems and in different perturbations. So I would say the way to avoid it is to not rely on the molecular alone, but to tie the molecular in with the phenotypic response and in perturbations.
I will triple that, I guess, because in the discussion comparing the different sessions, the frontiers, and one beauty of the immune system is that in a way the function is quite clear. I mean, it's really there to fight infection. And in a way, the outcome is extremely all or none and black and white in its outcome. So it gives us a very objective function to optimize. So we can work at the molecular level, we can work at cellular level, but at the end of the day, it has to tie up to something which is fairly rapidly decided, except in the very slow growing infection and HIV and maybe cancer. But in most cases, we are dealing with humans or a mouse who is going to make a decision within 1 week, is going to be able to clear the infection or die. And that's really the function we are trying to optimize. And you know, you have to make it in the context of not destroying your own tissue, not getting autoimmune disorders, but there is something which, on the big scale, at the physiological level, that we can really optimize against, and that maybe makes the modeling easier to carry out, I guess.
I think it's a really good question. I think that the one of the keys is setting up a way of testing models that's truly, if you have a precise quantitative model, that your evaluation of how that model is doing has to be a precise quantitative test. It shouldn't be a very qualitative, oh yeah, this whole colony's I'm predicting this whole colony's going to die. Well, you can cheat in a million ways if that's your outcome, right? If it's a digital outcome, if you have a precise quantitative outcome and you have a truly well set up test, that's then you can't fix the system, right? You know, in our case, we work a lot on the clinical side, and there it's probably even simpler to set up what the test is, which is it's got to be a clinical test where there's another independent method of assessing the whatever clinical parameter you're trying to test. If we're trying to say we can diagnose disease X, we need to compare to whatever another standard on how to diagnose that disease and see how we do.
I think I can use this one. So I guess we're all answering it in a slightly different context. But I'm thinking about the context of cell subsets. So I see a lot of data, trying to parse, as I alluded to, I'm interested in macrophages and their macrophage subsets. And you can look for all the different immune cells, parse it with all the single cell data more and more down, and use your statistical models to come up with different populations or clusters. And I think at the end of the day, how do we really know when we're at the right one, and is it important? And so it gets back to the same thing. So different contexts, but I think the same answer is, are there functional assays to look for whether these cell types are actually important? Do we see similar subsets emerging across different experimental systems and different contexts, maybe different disease in different organisms? So again, yeah, I think it gets back to always going back to the experimental data to have something to ground it in.
So I agree with everything everyone said, and so I'll try and add some other points. So I think, first of all, basically, this community is divided into two, or immunology. There's systems level people which are highly represented here, and I think are basically, if you look at, we're generating data, high-dimensional data. This has become now beyond systems level. Immunologists generate data, high-dimensional as well. And I think, first of all, what's missing in the majority of data, and I kind of alluded to this in my talk, is that it's underanalyzed, and the statistical analysis of it is actually quite poor. This is probably not representative of systems level people, but is representative of the majority of data that's coming out there, and the insights that we're seeing. The models are not correct. The prediction, the functional outcomes that people are looking for often are not there. People are generating single cell gene expression data and defining cell subsets on mRNA, which 15 years ago would have been considered blasphemy. So that needs to change, and it's a maturing field, and that will happen.
Then are you saying that what's been published so far is mostly wish fulfillment?
I think what's been published is a lot of observational data Most high-dimensional data that people are publishing is observational. Most of it does not have that functional validation that was raised here. And the majority of it is poorly analyzed or underanalyzed. That is going to change over time. So feel safe. The other side of this is that I think we need to, as a field, understand that the way that we do experiments in high-dimensional data, and particularly when we move to humans, need to change drastically. Bree mentioned the collaborative cross mice. This is a wonderful, so if you're talking about a system that has variation, and you're trying to understand the relationship between them, you can't do the experiments the way you did before when you did a knockout and tried to understand, just looked at the genes. You need to understand the relationships. You need that diversity. Those resources are, of little use right now. Very few people are using them. They're expensive. Very few people are measuring in high dimensional and looking at all the relationships. So that, again, is a maturing field. The training is going to come and it will change.
I will follow up on that. There is, in a way, a revolution in the field, which is that everything we know in immunology was based on genetic models, knockouts, and removing one component. And obviously, we learned a lot, and we could navigate a little bit of the system. But keeping the system in place, not perturbing it, and looking at the real dynamics of the system and the real function is a game changer. We're not asking what's necessary to build an immune response. We're asking what's sufficient. And in a way, it's drastically different in terms of method. It's coming about slowly, and maybe science evolves one death at a time. But I think the new generation of immunologists really catch this kind of epistemological difference of probing the system.
So a lot of these perturbation studies and more detailed studies are done in model systems of one kind or another, or in ex vivo settings, right? Do people feel that that can translate to human settings directly?
I mean, I think one way is Peter Sorger touched on yesterday, using these new high-dimensional imaging approaches, where in clinical samples then you're able to at least have an endpoint assay and look at the tissues and say, are the cell types that people are talking about actually present around a tumor or at a site of infection, right? So it's not to the same level that you can use an animal model, but you can still look to see, is the thing I predict to be there actually there, and what is the context around it?
I think the answer is sometimes, but we don't know how to distinguish the sometimes from the no, right? And I think that's why, I don't know, I can't even imagine how many billions and billions of dollars are wasted in the drug development world based on taking drugs that work in mice and then don't work in humans, especially in the immune system. I think is a particularly good example. I think the mouse and human immune system are vastly different, probably much more different than other systems that seem to share similarity. So I think when we were saying that we learned a lot from knockouts, we also learned a lot. It was also done in mouse models. I think a lot of immunology was learned in mouse and doesn't extend all that well to humans, and that's been the biggest revolution in the last 5 years. Is we've really have tools to look at the human immune system, and I think we're starting to at least pull out some of the. I think there's a whole bunch of people who made their careers just out of saying, "Hey, this doesn't work in this was dogma in the field, but it was based on mouse and doesn't extend to humans." So I think we're learning a lot as we go on that.
So I touched on it in my talk. I think basically what we need to do for all of these issues is put in those generalizable quantitative frameworks that let us look at differences. So mice are not human. We thought that for many years that we can extrapolate, but now we have the data to start going at it. We did like a first pass at this. Can get it much more sophisticated, right? And so if you're looking at this, you're putting the data in a framework that allows you to start making predictions on every different piece. You're going to start bridging some of those gaps. Simple model increases 50% the overlap, and it can get way more sophisticated over time.
So I agree with you, Shai. But another component I see actually as necessary is to include models that translate those translations from mouse to human actually into differences with regard to the behavior of the signaling networks, for example, which could be done, if we would invest into that. So if we take this translation map in expression levels and then feed it into somewhat mechanistic models of what it means, then yes, this translation could be done automatically, essentially. Yes.
So the issue of simulation modeling versus data analysis keeps coming up. It's kept coming up over the last day and a half. We'd be remiss if we don't discuss it today, I guess. So at one extreme, you could say that emergent phenomena can only be explained by integrative simulation models, right? At another extreme, one might say that if we collect enough big data, if data is big enough, we could explain phenomena directly from the data, and we don't need an underlying simulation model for it. Would anybody like to comment on that?
In a way, that's what's happening in the field right now in the clinics, right? We are running a lot of clinical trials in immunotherapies and such, which are really like practically experiments to prove the system. And of course, we have to do data analysis and machine learning to try to compress the diversity. But at the same time, we are changing the priors as we go, right? I mean, we are really putting new drugs inside the system, and without a model, we are never going to be able to anticipate and to design the next perturbation. So we need data analysis, machine learning to compress, to help us look at what are the main dimensions where things can be varied. But we still need the mechanistic model if we want to be able to predict the next perturbations and deal with the diversity of patients and parameters we didn't anticipate. And it's not perfect, but I think we are getting there.
Well, I want to just add to this. If you look at the total number of humans who are alive today, it's something like 10% of the total humans who have ever lived, right? So in a few more decades, we might actually be a bigger proportion of the total number of humans who have ever lived. And if you collected enough data with enough granularity, if you just let Google and Facebook and so on collect more data on us, will we still need mechanistic models?
The explosion of combinatorials with drugs, right? I mean, we are never going to be able to deal with it if we don't have a model, right? We need to go beyond just acquiring bigger data sets. We have to be able to predict.
And I think also, there's a tendency to confuse the ability to predict some outcome with the ability to understand where it comes from. You can train some machine learning process with a lot of data, and then it will say, okay, so this patient will go on to do this, and another patient will be like that. But in the end, if you want to interfere with it, if you not just want to watch the patients actually go along those predicted paths, but you want to interfere, you need, as Grégoire said, you need a mechanistic model, you need a molecular handle. So how do I stop this if it's negative? So I think we will always need the mechanistic models.
So I just want to say, to be clear, I'm a very strong supporter of mechanistic models. I just think, but I just think to do this for the scale of the immune system and really model what's happening in humans, we can't do it, basically one cell at a time, and with the way that we're trying to do it, say, in E. coli. And in fact, we need to actually take this very large community that is only generating the data and trying to make observational insights and funnel it so that it feeds into what our mechanistic models. They're not going to be as beautiful as the ones Martin is doing right now. You're going to have to compromise some stuff, but they're going to change the field that they have a very large impact because a huge amount of people actually generate that data, and it's going to, you'll give them, they move from 5% understanding of the data to 10% on a very large community, a very large amount of data. That's going to be a huge benefit. It's also going to support the modeling efforts, and that's going to eventually will get to every cell the way Martin does them.
No, eventually it will be there, Yeah. If you ask people in 50 years, will we be able to, simulate an entire liver or so with its functionality? I guess most people would say yes. On the other hand, still the opinion is, oh no, it's far too complicated, but eventually we'll be there, but we have to start somewhere. Yeah.
So you see simulation then in that context as a sort of culmination of data analysis? The crowning?
Yeah, I think we need models and we need mechanisms to understand the world. You could potentially think that, by generating the data, as you said, we may not need models. I think the problem is that, we're not going to be measuring everything, and measurements have noise and so forth, and besides, it's less fun. You want to understand the science, you need to actually get the mechanism. So.
Yeah, but you're right. Hybrid models, I guess, is an intermediate step, which will be very useful, yeah.
What about the question of scale? As I said at the beginning, the human immune system is just ridiculously complex, right? Trillions of cells, many dozens of interaction mechanisms and so on. is it even feasible to think of, large-scale models in immunology in general, or are we talking always about very specific simulation models, capturing specific properties?
I guess incremental, you can do it. I mean, this hybrid approach that we keep coming back to, where you have parts of your model being very phenomenological, high level, and then you say, okay, so for example, I observe that deficiency in the common gamma chain or whatever in some element leads to a certain disease, and then you want to understand how, and the answer obviously is somewhere also at the cellular level, and for selected problems you can then perform very detailed mechanistic models and essentially plug them together wherever you see the need, and try to build some hybrid where, as I said, for some aspects high level and for some aspects very detailed models are being used.
Oh, and I was also thinking that at least in the immune system too, with the imaging techniques that are being advanced so rapidly, and the accessibility of the cells in the immune system. I mean, yes, the models are very complex, but at least we can test them against things in vivo, where that's where the complexity is, and that's where we want to model. And so I think that is, makes the immune system actually a great test case for whether this is even possible, maybe, to build these up. Because we actually have the tools and we can access some of that complexity and have something to fit it to.
I think the big problem and the difficulty in doing modeling the system is that, I think trying to go after the whole thing, as you were saying, is probably somewhat of a fool's errand to start with. It's just a massive system with so many parameters that even if we collected data, massive scale data, for what we call massive scale data for the next 1,000 years, it still isn't going to come close to establishing what those parameters are supposed to be. So you have to subdivide it into smaller problems. But I don't think we've yet, or at least not that I'm aware of, figured out how to decouple whatever one's looking at from the rest of the system. And like in physics, you have an energy scale where you can say, okay, the gravitational forces are much weaker than electromagnetic forces. So if you're studying an electromagnetic problem, you can literally ignore gravity, and what parts of the model you could ignore. In biology, there's no concept of that. So oftentimes people pick a subset to look at, but they just look at that because that's what they can either deal with, or because they those are the things interesting to them. But if they can't, there's no parameter where they can show the rest of the system decouples. So just because you're ignoring it doesn't mean it's not there and impacts your model. So until we figure out how to do that, I think we're going to be a little bit stuck. But as soon as we do figure out how to decouple that, and maybe there's really good specific questions where we can think about how they decouple, then we can probably have, with the massive amount of data we are that creating and have a good model, we can intersect the two and probably chip away at really good smaller problems.
Yeah, I will expand on that, because we have examples in solid state physics when people learn how to renormalize system, right? To really start from microscopic things to be able to learn how to move from one scale to another, and in a way, in these simple Ising models and solid state physics, people could do that. It has been very successful, obviously. In the case of biology, and that's what Arlen is pointing out, is that there might not be separation of scales. And in a way, it's a very nice theoretical challenge to really understand how we go from the molecular to the cellular to really the systems. And the lack of decoupling of these time scales is maybe the beauty of the immune system. It can do that on a big scale and really match what it cares in terms of physiology, in terms of protecting the tissue, in terms of rejecting the tumors with the molecular scales, and be able to go from this very short time scale to this very long time scale to the very individual molecular events to the whole scale of the organisms. And that's what is a good theoretical challenge, I think, for a lot of people.
But then if you consider those issues, I mean, think about the stiff-arm pendulum, right? With the stiff-arm pendulum, you have perfectly predictable behavior. You introduce one break into the arm so it can bend, right? And you have perfectly unpredictable behavior. So one little change in our model can make a huge difference to the system.
Well, it can't be, the whole system cannot be wired like that, right? There are going to be parameters where you don't get the right TCR, you don't get exactly the right amount of cytokines, and you are messed up and you die. But obviously the system is quite robust, so we don't have to care about every single molecular reactions. No.
I agree. I think there's a lot of evidence, actually outside the immune system, even beginning with yeast and so forth, E. coli, but certainly with the immune system, and you're looking at all these variation that exist, that actually a relatively small amount of things are actually the key factors. What they are exactly by context actually varies different, and we don't know them in full, but it just look at, from a data-driven perspective, look at individuals where you're profiling them longitudinally. They're going to look very different from one another. You're going to take the same person over time. Very few things actually change in a short time scale, and you can immediately zero in on what are the more important things. And if you're looking at them, over we've done it years, you can see very clear dynamics of what's going on. So, and from that comes hypotheses for mechanisms. So, we think of it as a very complex system. I think a lot of it. There was a few examples that were given throughout the symposium, but there's a lot of constraints that we don't yet fully understand. And once when we do, I think some people talked here about essentially geographical constraints in tissues and so forth. That drops down the level of complexity to a large extent. So some hope, I think.
I'm playing devil's advocate here. There's also an argument to say evolvability necessarily requires modularity, and so we will find modules, right? So let me change tack. I want to ask another question that we, the Immunology Institute, are discussing daily, which is how do we define healthy humans? What's a healthy immune system? Is there a single healthy immune system? Is it multiple distributions? Because we're comparing, we talk about healthy controls and disease, right? But there are genetic factors, predisposition factors, for example, environmental factors, aging, all kinds of things that change things, right? And I know you've been collecting a lot of data on this, so I'm curious to know what everybody thinks on the topic.
I'm not actually a card-carrying immunologist, but I will. I think it's context specific, right? And I think actually defining what healthy immunity is has changed drastically. And if we look at the general chronic illness that we have as a community, where most of us are in a state of more inflammation than we used to have, I think actually defining what a healthy immune response has to be functionally defined, which is in the context of your environment. Does your own immune system, is it at the correct resting state? And does it respond appropriately to perturbation?
So if you live in an environment where TB is not a problem, then you're healthy even if you're predisposed to TB?
Well, there's everything else. So actually, it's an interesting question. So there's this question about BCG, which is the TB vaccine, and it works in places like Northern Europe, where there aren't a lot of environmental mycobacteria. So BCG vaccine gives people exposure to mycobacteria where there were no environmental bacteria. But around the equator, where we have TB disease, the BCG vaccine doesn't work well for adults on respiratory disease. And the thinking is that there's a lot more environmental mycobacteria. So the resting state of what the immune system has, how it's already responded to mycobacteria, is brought up to the point where the BCG vaccine doesn't work at all.
This ends up being an extremely complicated question, and there's certain areas where we get some test beds to start looking at this in humans. So in the case of, Fred Hutch's kind of clinical claim to fame is bone marrow transplants, right? And there you kind of reset the immune system, and then the big question clinically is, okay, so you have a new immune system for each of these people, and we have these highly toxic drugs, prophylactic drugs that you have to give these people for basically the rest of their life, but certainly you dose it according to how well you think their immune system's doing. So it's a very practical question, right? At what point you want to stop or reduce the drugs if their immune system's working. So how do you know if their immune system's working? And so, it's a good test bed because we know when it's not working, in the sense that at least in certain ways. I mean, if people at the extreme end, significant enough people die of infection, where there you pretty sure that the that normal, other people don't die of, fungal infections. So for example, for sure that on that extreme end, you have people whose immune system is not functioning properly. And then you have groups that seem to be completely fine. So you can at least get the middle's fuzzy, but you can at least get some groups that you can start probing and saying, okay, what measurements really seem to correlate with one versus the other. But I think we're a long way from nailing down an understanding of what the whole system is that has to work together. We can look at individual things that seem to correlate with it, but it's a hard question.
If I can take a stab. So basically that data set that we've been tracking, which I think at this point is the richest human data, immune monitoring data. It's right now 12 years of data on many, many different high-dimensional measurements, defines what I think is a healthy immune system, or what the pattern by which people change in Palo Alto. It's a very small claim, but I know what people in Palo Alto are going to, what their immune system looks like in year X, and I can predict how they're going to look like, at what rate they're going to change, and so forth the following year. I also know that this, I didn't get a chance to show, it correlates with cytokine responses, so functional readout, and it has a clinical implication. So, array, a bit small, small achievement. Now, you go to all these other places, as Harlan and Bree mentioned, we have one pattern. Does it mean that that pattern is going to replicate in the next to the equator, or is it a different way of what people are changing, what's considered health? I don't know. We're starting those studies now. But I think, and at least we have some preliminary data, that actually knowing very well one such trajectory, one such change of how people kind of move along an immunological landscape is really helpful in doing this much easier in other contexts. So I don't need to measure 12 years of data now to start taking people and putting them on this trajectory and assess their information. So I think the jury's still out, but it's one step forward. And I think any such data sets that we generate are really high-dimensional, longitudinal, are going to give us some of those answers, and we can start skipping between data sets.
Just add that people from Palo Alto are obviously not normal. But
Not normal. I definitely agree.
Okay. Let me, just before we finish, I want to ask you each to say, if you would, just briefly, is there one thing that you think would be the next best thing to happen in immunology research from your perspective? Where would you like the field to go next?
Well, I think we discussed it a couple of times. Not directly responding to your question, but the ability to bridge data and insights at different levels, to develop some kind of an approach where you can combine very low-level mechanistic models with population scale models, and perhaps in a way that you can actually quantitatively assess how well your model is doing, which is, I think, very difficult. But that's, I guess, one of the big challenges and would be a great achievement.
Bree?
I think for the TB field, it actually mirrors perfectly this theme of data integration. So we have this major question of how do you develop a vaccine, because we can't identify the difference between individuals that will respond to different perturbations. So we can't tell you if you have latent TB. There are no consistent biomarkers that will anticipate an individual who will just have latent disease with no problem versus individuals that progress to active disease. And we don't understand the difference in the immunology in the immunological state of individuals that work in TB clinics that never get disease, right? They're clearly exposed all the time, so they're super non-responders. So there's been a lot of emphasis in starting to measure all these biomarkers, do the genetic studies, and now hopefully follow along with the phenotypic studies. And we still need a way to quantitatively link these time and measurement scales together.
I completely agree.
Come on.
The genetic diversity, the fact that every tumor is different, what it means in terms of the functioning of the immune system, mapping the TCR, BCR repertoire, the genotype of the tumors, and being able to predict on a long-term scale is fascinating things we need to go after, I think.
I would really like to see the field progress more in terms of immune tolerance, right? We put a ton of money into cancer research where the concept of what's going on makes a lot of sense, and yet but things like autoimmune disease gets far smaller amounts of research, and our understanding is really quite poor in terms of what's actually going on, and yet it's obviously some of the most common diseases in the world, and certainly the biggest drugs in the world, selling drugs in the world are for autoimmune disease. Yet they're just basically anti-inflammatories. There's nothing about the disease that shows they're not connected to the disease at all. It's like where cancer drugs were 20 years ago or 30 years ago, it's where the autoimmune drugs are today. And I think we could really make a huge dent if we could really start understanding what it means to attack self and how we tolerize to prevent that.
Can I jump in really briefly to add on to that? Sorry, Catherine. That I think we're missing a lot of information about the role of metabolic state and immunity, and we're starting to see that play out in TB, but across not only infectious diseases but chronic diseases as well. And I think it's just an area that they're just being separately studied when we know they're tightly linked together, and we have no idea how.
So I definitely agree with that, and I keep thinking I need to read up on metabolism. But also, kind of related, I was going to bring up a sort of softer point that might relate to that. Is okay. So recently, I've gone to Cold Spring Harbor a lot for immunology meetings. When you go to the immunology meeting for gene expression and signaling, there's the T cell session, and there's the B cell session, and there's the innate immunity session. And it's great. I mean, it's great. They go deep, and they understand that part. And yet here what we're talking about is the emergent behavior of all these things working together. So then we organize the systems immunology. And there, there's sort of, signaling in all these different systems, or sort of repertoire and across different systems. But we need to integrate our data, and we also need to integrate our people, I guess. And that one, then there's metabolism, and there's the neuroscience. And so, I mean, I think at some point, we're just going to, and I don't have an answer for this, but at some point, we are going to have to figure out how to kind of effectively bring us together and have a similar vocabulary to talk about these things. And that will really advance things, yeah.
We need a section.
So actually, to follow, it's really the same point. I think what this community needs the most, and I saw this yesterday with the folks in neuroscience talked, and I was, somebody said 10 individuals could fill, we can't fill Australia. I think we can't even do Luxembourg. The basic idea is that at this point, unfortunately, I think immunologists and system level kind of people and as you said in conferences, are not really conversing. In fact, it's an alien thing. You don't necessarily read the system level papers. And many people who are doing kind of system level analysis are not necessarily bringing their papers to the level that would bring in the interest of the immunologist and pull them in. That needs to change really for the field to advance. The other side, I guess, of what you said in terms of the sessions, Catherine, is that look, any given data measurement that we do now, if I run CyTOF and I'm a B cell expert, the number of markers I have, I'm actually probably running and I'm getting information on all these other cell subsets and forth, which I'm not an expert. I can't leverage the data. I can't do anything with it. I usually, would chuck it or won't be able to understand the relationship. If your measurement tools are essentially across the domain, we need to build all those tools to let us leverage the entire domain from any one experiment.
We can't run away from this problem, right? If you do site-off, you do get these other cell types, and I think people are going to be forced to deal with it, right?
No, this is exactly why we built this natural language processing engine, just for the fact that when you get a result, that you would minimally get all the information in from all the other cells that you're not an expert on, right? It needs to be. The system, no cell here works in isolation.
I want to thank you all for very inspiring thoughts, and thank you, the organizers.